Today I'm going to discuss on oxidation of short chain fatty acids, peroxisomal oxidation of long chain fatty acids, and alpha and omega oxidations. Oxidation of short chain fatty acid. The short chain fatty acids are oxidized in the same manner as the even chain fatty acids. At the end of final oxidation cycle, the 3 carbon fragment propionyl coenzyme A is left behind instead of acetyl coenzyme A, which happens in case of even chain fatty acid beta oxidation. The propionyl coenzyme A is further metabolized to succinyl coenzyme A, which enters the TCA cycle. So this is the importance of this odd chain fatty acid beta oxidation. The end product is that is the propionyl coenzyme A is able to be converted to succinyl coenzyme A, which is an important intermediate of Krebs cycle. So we'll look at here in the steps of conversion of propionyl coenzyme A to succinyl coenzyme A. So propionyl coenzyme A, in the presence of enzyme propionyl coenzyme A carboxylase, whenever the name of carboxylase comes, always the coenzyme biotin is there. ATP, 1 ATP is utilized and ATP is converted to AMP, that is equal to the conversion of, that is equal to the absorption and utilization of 2 ATPs, because 2 high energy bonds are being broken. And then this propionyl coenzyme is being converted up into 3-methylmalonyl coenzyme A. So the carboxylic group is added here in the middle carbon, so it becomes chiral. Now the D-methylmalonic coenzyme A in the presence of enzyme epimerase is converted up into L-methylmalonyl coenzyme A. Simple epimerization is there, flipping at the chiral carbon leads to L-methylmalonyl coenzyme A. So in the presence of methylmalonyl coenzyme A mutase enzyme, and vitamin B12 is also required as a coenzyme. This entity is converted up into the succinyl coenzyme A by simple shifting of this carboxylic group from the second position to the third position. So this is the structure of succinyl coenzyme A. Now, what is the clinical importance of this oxidation? Short chain fatty acid oxidation, it is due to the defect in the enzyme methylmalonyl coenzyme A mutase or deficiency of vitamin B12. So suppose this enzyme is absent or deficient, so what will happen? There won't be any conversion of L-methylmalonyl coenzyme A to succinyl coenzyme A. So there will be severe accumulation of methylmalonyl coenzyme A in the body, and followed by its excretion in the urine. Start excreting into the urine. It causes severe acidosis, damage to central nervous system, which retards the growth and development. So this is the effect of methylmalonic acidemia due to the absence of either vitamin B12 or methylmalonyl coenzyme A. Now, beta oxidation in peroxisome. So, beta oxidation occurs in the modified form in the peroxisome. The FADH2, which are synthesized in beta oxidation by acyl coenzyme A dehydrogenase enzyme step. So, they are not transferred to electron transport chain but handed over to the oxygen. So, this result in the formation of H2O2, and there is no ATP production in the peroxisomal beta oxidation. It is now believed that peroxisomes carry out the initial oxidation of long chain fatty acids, which is followed by mitochondrial oxidation. So peroxisomal oxidation is induced by high fat diet and administration of hypolipidemic drugs. So it is an energy conservative process. FADH2 is not going to generate the energy through electron transport chain, as the peroxisome lack the that ETC system. So we can see this is a long chain fatty acid. So it is going to be converted up into the trans-enoyl coenzyme A by simple dehydration at this step. FAD is utilized and converts up into the FADH2, and this FADH2, what used to be the fate of FADH2 in the normal beta oxidation, that goes to the electron transport chain, and it's converted up into, it generates a 1.5 ATP. But here this is not the case. This is the major difference, and this FADH2 will have to revert back to FAD. And in course of this conversion, the water molecule is converted up into the so water is converted up into the H2O2. In the presence of enzyme catalase, again it can revert back to water, and this cycle goes and goes on. So this is the major difference. Once this trans enol coenzyme A is formed, so there is no further oxidate, no further conversion or no further steps of beta oxidation happens in the peroxisome. Rather, this product will shifted to the it's shifted to the other sites, to the normal cells for a normal mitochondrial beta oxidation. So this is the importance of beta oxidation of long chain fatty acids in the peroxisome. Now, what is the clinical importance? Zellweger syndrome is a rare disorder characterized by the absence of peroxisome in the tissues. As a result, the very long chain fatty acids are not able to be oxidized, and they keep accumulating in the various tissues like brain, liver, and kidneys. Hence, this disorder is known as cerebrohepatorenal syndrome. Right. So Zellweger syndrome is associated with absence or deficiency of peroxisome, where the long chain fatty acids are not oxidized, they get accumulated to the brain, liver, and kidneys. Now, alpha oxidation of fatty acid. This is also a minor pathway for oxidation of fatty acids, and oxidation occur at the alpha carbon atom. So one carbon is removed from the carboxylic end of fatty acid as carbon dioxide. So it does not require coenzyme A and does not generate ATP. This process occur in brain and nervous tissues. So you can see it here. This is phytanic acid. This is a kind of fatty acid which has got a whose beta position is blocked, because at the beta position the carbon is loaded with another methyl group. So beta position is blocked, so there is no beta oxidation. Rather, it prefers to undergo alpha oxidation. So what will happen? Phytanic acid may undergo hydroxylation. So under the influence of enzyme monooxygenase family, there is a phytanic acid hydroxylase enzyme. In the presence of oxygen and iron and vitamin C, there is a addition of hydroxy group at the alpha carbon atom. So this is L-hydroxy fatty acid being formed. Further, the dehydrogenase enzyme converts this hydroxy group into the ketonic group, and with subsequent loss of carbon dioxide, and under the influence of enzyme decarboxylase. So this is how like the process happens. So this deficiency of the enzyme, this phytanic acid hydroxylase, will lead to there is no conversion of phytanic acid, there is no oxidation of phytanic acid, it keeps on accumulating. So Refsum's disease, it is a rare but severe neurological disorder characterized by cerebral ataxia and peripheral neuropathy. The affected patients show accumulation of fatty acids, especially the phytanic acid, which is abundantly present in green leafy vegetables and the milk. It is caused by a defect in the alpha oxidation due to the deficiency of enzyme phytanic acid hydroxylase. This enzyme is lacking, which is required for hydroxylation of phytanic acid. So which is derived from dietary chlorophyll by oxidation. So phytanic acid contains a methyl group on the C3 or the beta carbon that blocks the beta oxidation. So this is the overall, account of alpha oxidation of fatty acids and its clinical importance. Now, omega oxidation. In the omega oxidation, it is also a minor pathway for oxidation of fatty acid. It involves the hydroxylation followed by oxidation of omega carbon atom of the fatty acid. The reaction takes place in the endoplasmic reticulum and requires the cytochrome P450 and NADPH. So what is happening here? This is the omega carbon. Suppose this is the omega carbon. So under the influence of cytochrome P450 hydroxylase enzyme, there is a hydroxylation at the termini at the omega methyl group, and it converts up into some kind of dicarboxylic acid. So this is the account of omega oxidation.